Hi all, I am Christos Frantzidis and I will give the first 5 lectures within the module of neural computing. So this week we will investigate the basics of neural computing, giving an introduction to what it is, the module, and the basic neuron structure. So this is my email, and you may contact me directly either through email or coming to my office. First of all, what is neural computing or computational neuroscience? It is a relatively new field that combines uh, concepts from neuroscience and fuses them with computer science, mathematics, and physics. Its objective is to understand how the brain processes information. For example, to understand how uh, vision occurs in the human brain or how we can remember things. What does it involve? We will speak a lot of, especially during the next lectures, about models. These are mathematical models, uh, which may be something like that, a formula, and we will speak also for simulations and algorithms which try to represent and analyze the complex functions of the human nervous system. This may start from a basic neuron structure and how it communicates with other neurons until we may have more complex models like neural networks or even the entire human brain. But to be able to understand how the brain works, which is here a very complex system, we should start from the basics. And what do we mean as basics? We will start from examining the neuron, which is the cell of the central nervous system, and its basic structure and how it communicates. So, what is the basic, sorry for that, what is the basic neuron structure? You see that it has a relatively complex shape. This one here, it is called the cell body or soma. Soma is a Greek word. This specific circular parting at the center of the cell body is the nucleus, which is very important. It is the core of the nervous system. You see here that long prolongation, which is the axon. We may see also here these components, which is the myelin sheath, and are used for insulation and transmitting the information much faster. You see that this insulation is not continuous, but there are some gaps that are called the nodes of Ranvier. Finally, we have the axon terminals, which is the end points, and there are some tiny um, prolongation which are the synapses. Okay, this is the basic neuron structure. Let's start from the cell body or soma. You may see here at a zoom how a cell body looks like. It is the central part of the neuron which contains the nucleus, the pink one, and most of the cell's organelles. It is responsible to maintain the cell's health and metabolism. It is like the brain of the nervous of the neuron, since it integrates the signals received from the dendrites. Dendrites are the inputs to the neuron. The cell body may generate an action potential, an electrical signal, if the input signals are strong enough. Now, regarding the nucleus, it is important because it contains the neuron's DNA. The nucleus controls the neuron's activities, including protein synthesis, and the cytoplasm, this part, contains typical cell organelles, the most important of which are the mitochondria, which are crucial for energy production. Apart from the mitochondria, it also contains other organelles like the endoplasmatic reticulum and Golgi apparatus, which are involved in cellular processes like energy metabolism and protein production. Okay. Then we have the dendrites. These are tree-like extensions from the neuron's cell body. This one. They receive signals from other neurons or sensory receptors. The dendrites act as the input region, collecting information that will be sent to the soma, to the cell body. They have many branches which allows them to receive signals from multiple neurons at once. This is the complexity of the human nervous system. These signals that dendrites receive are usually in the form of neurotransmitters binding to receptors on the dendrites. Sorry, the dendrite surface, leading to small electrical changes in the neuron. Okay. Now let's go to the axon. The axon is the long, thin extension, that one, that transmits electrical signals. We will see that we call them as action potentials, away from the cell body towards other neurons or effectors, muscles, glands. So the axon functions as the neuron's output pathway. The axon hillock, that one, is the region where the axon connects to the cell body. The axon hillock is the point where the action potentials are generated if the incoming signals from the dendrites and soma reach a certain threshold. Now, the axon, the synaptic terminals, is the end here of the axon branches where neurotransmitters are released to communicate with target cells, which are other neurons, muscles, or glands. Then we have the myelin sheath. This is the insulating layer that covers the axon. It allows faster transmission of electrical signals. It helps to prevent signal loss and increases the speed at which action potentials propagate along the axon. Myelin sheath is made of glial cells, specifically Schwann cells in the peripheral nervous system and oligodendrites in the central nervous system. The myelin sheath is not continuous. As we saw, there are small gaps called nodes of Ravier. What are the nodes of Ravier? These are small gaps between segments of the myelin sheath. They play a key role in saltatory conduction, which allows action potentials to jump from one node of Ravier to the next. This greatly increases the speed of signal transmission, and the nodes of Ravier are structured as bare regions of the axon membrane where ion channels are concentrated, enabling the rapid propagation of action potentials. This is the case derived from Encyclopedia Britannica, where we have a myelinated axon and here an unmyelinated. The velocity, the speed of action potential propagation is faster in the case of the myelinated axon. Now, how the neurons communicate. We saw that the end part of the axon are the axon terminals. These are small branches at the axon's end, each with a bulb-like ending called a synaptic knob or button, which contains vesicles filled with neurotransmitters. At the end of the axon terminals form connection synapses with other neurons, muscles, or glands. When an action potential reaches the terminal, it triggers the release of neurotransmitters into the synaptic cleft, which then binds to receptors on the postsynaptic cell, influencing its activity. Okay. Then we have the synapse. The synapse is a point of communication between two neurons or between a neuron and a muscle or gland. It allows the transmission of signals from one cell to the next one. The presynaptic neuron is the one which sends the signal and releases the neurotransmitters. And we have the postsynaptic neuron, which is the one that receives the signal, which contains receptors for the neurotransmitters. So we have two neurons, the one who sends, the presynaptic, and the one which receives, the postsynaptic. The synaptic cleft is that part, is the small gap between the presynaptic and postsynaptic cells through which neurotransmitters diffuse. Okay. Now, we learned about the basic neuron structure. Let's come a bit earlier to the point that we made the introduction to neural computing and see what is the goal of neural computing. It is to understand brain function in terms of its underlying physical and biological mechanisms and to predict the behavior of neural systems under various conditions. What do we mean as brain function? We mentioned earlier the problem of vision. It is very important. You see that vision is typically produced in the posterior part of the brain, which is the occipital cortex. but we may also discuss with sensory informations and sensations, which is the central part, the somatosensory. We may have memory, which is the temporal cortex, consciousness and balance, which are subliving regions, speech, which is frontotemporal cortex, and movement personality, which is more on the anterior part, the prefrontal cortex. And in parallel to the brain, we have an artificial neural network, which again receives an input. Here it makes some processing through hidden layers, for example, and produces an output. Now, let's start talking about how the neurons communicate and the signals that they produce. Firstly, we will start from the resting membrane potential. What is the resting membrane potential? It refers to the electrical charge difference across the membrane of a neuron when it is not actively sending signals. In that case, it is like the neuron is in a resting state. In most neurons, that resting potential is a bit negative at around minus 70 millivolts. Two key ions involved in setting the resting membrane potential are the potassium. We have higher concentration of potassium inside the neurons. It is the green ones. And we have higher concentration of sodium outside the neurons, the red ones. This does not mean that there are no sodiums inside the cell or potassiums outside. I cannot find here that one. Also, you see that outside there are more chloride negative ions rather than inside. So this is the case that it is outside, it's more negative than inside, so the difference outside minus inside is a negative. We will see both these two ions, potassium and sodium, a lot of in the next sessions. And since these are important and we should remember what happens with sodium and potassium, I would give you an example of a salty banana. I would like to remember the neuron in rest as a salty banana. This means that inside there is the banana, potassium, and outside is the salt, which is the sodium. If you remember that figure, you can remember what is happening inside and outside the neuron. Now we have a voltage difference, minus plus. This membrane potential, this difference in voltage, exists because there are different concentrations of ions such as sodium, potassium, chloride and other ions inside and outside the neuron. However, this is not a completely steady procedure because the cell membrane is selectively permeable, allowing some ions to pass more easily than others. Now, let's see about the ion channels and the permeability. You may see here that there is a gate, a channel. This is a potassium channel, which the potassium channels in general are leaky, allowing some potassium ions to flow out of the cell. There are very few sodium channels open at rest, so sodium ions cannot enter the cell easily. So basically we have more potassium running outside than sodium entering the cell. As potassium ions leave the neuron through leak channel, they carry positive charge out of the cell. Initially we had 1 potassium, now we have 2. And so we have a voltage difference here that is creating. Since sodium ions are mostly kept outside the cell, this negative charge remains inside. This creates a chemical gradient from the inside to the outside. And as the potassium moves out, a separation in charge, this difference, is being created. So, as potassium runs outside the cell, there is a relatively negative charge on the inside, and this creates an electrical gradient. Okay. As more potassium leaves, the electrical charge is increasing, and eventually this starts a flow back in, and now there is potassium movement both inside and outside. The leak channels do not allow much of the potassium to flow out. I have put here a link that visualizes the simulation of this procedure. It is very interesting and fascinating. Please watch it. Now, apart from the potassium channels, we have sodium channels. Mostly, they are voltage sensitive and open in response to changes in the membrane potential. You may see here the potassium channel, which we examined before, and now there is a sodium channel. The sodium channels are typically closed at the resting membrane potential. They open when the membrane potential becomes more positive, depolarized. At rest, around -70 millivolts, the sodium channels prevent sodium ions from entering the neurons. Apart from the simulation, this is a very good explanatory video that you may watch, and I took the images from here. So now let's go and see the sodium potassium bump. If there was no, if this bump does not exist, we were just to let this go, and eventually we would lose that potential because all the sodium would come in. So we have the sodium potassium pump, which moves 3 sodium ions outside of the neuron and brings 2 potassium ions inside to the neuron. So it extracts sodium and attracts inside the potassium. However, the procedure of this pump is to move ions against their concentration gradients, and this requires a lot of energy, which comes from the adenosine triphosphate, or in abbreviation ATP. ATP is very important because it is the food of the cells. Now, why the sodium-potassium pump is so important? Firstly, it maintains the concentration gradients of sodium and potassium, which are crucial for the resting membrane potential and the generation of action potential. By keeping more sodium outside and more potassium inside the neuron, the pump ensures that the neuron stays polarized. This means the inside is more negative. Without this pump, the neuron would gradually lose its membrane potential as sodium would build up inside the cell and potassium would leak out. However, this pump is energy intensive, using up a significant portion of the ATP produced by the neuron. And this reflects how critical the ion balance is for proper neuron function. Okay, we saw the membrane potential, but now let's see how the action potential is being generated. What is the action potential? It is a rapid temporary change in the electrical membrane potential of a neuron or other excitable cell, such as a muscle cell, which allows it to transmit signals. It is the fundamental mechanism by which neurons communicate and send information over long distances. We have 5 steps which describe a detailed breakdown of what an action potential is and how it works. We see a typical action potential and the basic steps. Let's start decomposing the action potential. Now, before the action potential, the neuron is at its resting membrane potential, which we previously saw, and this is typically around -70 millivolts. This means that inside of the neuron is more negative than the outside. This resting potential is maintained by the sodium-potassium pump and leak channels, which maintain the concentration gradients of sodium and potassium ions across the membrane. We saw that there is more sodium outside the cell and more potassium inside the cell. When we have the case that an action potential is initiated when the membrane potential becomes less negative, is being depolarized to reach a threshold potential, usually around -55 millivolts. How that depolarization occurs? It can be triggered by inputs from other neurons or sensory stimuli that cause excitatory postsynaptic potentials. So from -70 we go to -55 millivolts. Once this threshold is reached, the voltage-gated sodium channels open and sodium ions rapidly flow into the neuron due to their concentration and electrical gradient. We see here that we have the influx of the positive sodium ions, which causes the membrane potential to rapidly become more positive, moving towards plus 30 or plus 40 millivolt. This is that part of the action potential. Shortly after the sodium channels open, they quickly become inactivated, stopping the influence, the input of the sodium ions. This is where we reach the peak of the action potential. At the same time, the voltage-gated potassium channels open and potassium ions flow out of the neuron, driven by their concentration gradient. So at that phase, we have the efflux of potassium, which brings the membrane potential back toward the negative resting level, which is known as repolarization. So the action potential starts to descend because we have a lot of potassium, which is a positive, to go outside. The potassium channels remain open for a short period after the neuron repolarizes, so it is not a completely simultaneous procedure, and it allows more potassium to infiltrate the cell than necessary. This causes the membrane potential to become more negative than the initial resting condition. So now we had in the beginning minus 70, and now we go to minus 80 millivolts. So this phase is called hyperpolarization or the after potential. Eventually, the potassium channels close here, and the sodium-potassium pump restores the neuron to its resting membrane potential. That part. Now, we have the refractory period, which is very important and is we have basically two periods, the absolute and the relative refractory period. The absolute refractory period happens during the depolarization and repolarization phase. During the absolute refractory period, the neuron cannot fire another action potential because the sodium channels are inactivated. The relative, no matter how large the input is, we do not have another action potential. During the relative refractory period, we may have a second action potential, but it is more difficult to be generated. The relative refractory period follows the absolute refractory period. The neuron can fire another action potential, but a stronger than normal stimulus is needed. And why that? Because the membrane is hyperpolarized. Now, it is very important, the absolute refractory period, because it ensures unidirectional propagation, discrete action potentials, and frequency control. Also, it prevents the backward movement of the action potential, ensuring proper signal transmission. Third, it sets an upper limit on firing frequency, enabling neurons to properly encode information through the modulation of action potential frequency. And it protects neurons from becoming overexcited or entering a state of continuous firing, which could lead to cellular damage or dysfunction. You may see here a figure generated and showing the absolute refractory period. Now, let's talk about the propagation of action potentials. The action potential does not remain in one place, but propagates along the length of the neuron junction. As the membrane potential is on a region, in one region of the action that depolarizes, it triggers adjacent voltage-gated sodium channels to open, propagating the action potential down the action towards the action terminals. This is the saltatory conduction, which greatly increases the speed of signal transmission. In myelinated neurons, the action potential jumps between the gaps in the myelin sheath, called the nodes of Ravier, as we showed earlier. Now, the theoretical part of this section finishes here, and we will try to see how this can translate it into models and simulations. But in order to be able to do that, we need a strong mathematical knowledge. Please do not be afraid, we will dive into those concepts gradually together. What we need is ordinary differential equations for describing voltage changes over time. We will see them in the workshop in more detail. We have also the partial differential equations for modeling signal propagation along an action. Thirdly, we need exponential functions for modeling ion channel dynamics, circuit theory for understanding the neuron's behavior as an RC circuit, electrical circuit, numerical methods like Runge and Kuta for solving the differential equations, probability theory for modeling stochastic ion channel behavior, Fourier transformations for analyzing neuronal responses to different frequencies, and conductance models like the Hodgkin and Huxley for ion channel behavior. Let's start by the ordinary differential equations for describing voltage changes over time in neurons. The membrane potential of a neuron changes over time as ions flow in and out of the cell. This is what we saw in the theoretical part. This dynamic change can be mathematically described by ordinary differential equations. The most common approach for modeling the voltage change in neurons is based on the principles of electrostatics and current balance. It is often conceptualized through the Hodgkin-Huxley model or its simplified derivatives like the leaky integrate and fire model. At the heart of these models is the equation governing the membrane potential. We will see it now. The voltage changes due to the flow of ions across the membrane through various channels. This movement of ions is captured by ordinary differential equations. So, let's see the current balance equation. The voltage change across the neuronal membrane can be described using the current balance equation. It is based on the law of conservation of charge and is described by Cm, which is the membrane capacitance measured in farads per square centimeter, The membrane capacitance represents the cell membrane's ability to store charge. We also have the derivative, which is the rate of charge of the membrane potential over time, and their multiplication is equals with the total ionic current flowing across the membrane. This is membrane capacitance multiplied by the rate of charge of the membrane potential over time equals to the total current, ionic current. Now, we say the total ionic current, but it is not a single entity. It is made up of different ionic currents flowing through channels specific to various ions. We have the sodium ions, the potassium ions, and leak currents that accounts for other small ions. Additionally, there may be an external input current which represents external stimuli applied to the neuron. So we have that the total current is equal with the externally applied current minus the summation of the three ionic currents. The external current is applied externally, as the name reveals, and it is due to a stimulus or an electrode. The sodium current is driven by sodium ions flowing through voltage-gated sodium channels. The potassium current is driven by potassium ions flowing through voltage-gated potassium channels. And we have the leak current, which represents the constant passive flow of ions through non-gated channels. Now, let's go to the current-voltage relationship or the Ohm's law. For each ion current, the relationship between the current and voltage can be described by a variation of Ohm's law. This law relates to the current to the conductance of the channel and the difference between the membrane potential and the ion's reversal or equilibrium potential. So for each current we have that the current equals the conductance of the ion multiplied by the voltage difference. The conductance of the ion channel Gm is measured in Siemens. It may be a function of time or voltage, especially in voltage-gated channels, and the reverse potential of that ion is the voltage at which there is no net flow of that ion across the membrane. For example, we have sodium current equals to the conductance of the sodium channel multiplied by the membrane potential minus the ion's reversal equilibrium potential for the sodium. Now, let's see how to build the model. There are 6 steps. First is to initialize the parameters. The membrane capacitance represents how easily the membrane stores charge. It is measured in microfarads per square centimeter. It affects how fast or slow the membrane potential changes in response to currents. Higher values of the membrane capacitance means that the membrane potential changes slower. Lower values lead to faster changes. The leak reversal potential is the voltage in millivolts at which the leak current reverses direction. This means that no net flow of ions occur through the leak channels. In this case, we set it as minus 54.4 millivolts. The leak current drives the membrane potential towards the value when no external current is applied. We have also the leak conductance GL, which is measured in millisiemens per square centimeter. It represents how easily ions leak across the membrane through passive non-voltage gated channels. Higher values in the leak conductance means ions flowing more easily, driving the membrane potential back towards the leak reversal potential faster. And the resting membrane potential is the baseline voltage of the neuron when no stimulation is applied. Here it is set to minus 65 millivolt, which is a typical value for many neurons. And you may see here the MATLAB code for initializing the parameters. Then let's go to the time parameters, to the time setup. Firstly, we define the time step, which is the time increment in milliseconds for each step of the simulation. Smaller time steps give more accurate simulations, but take longer to compute. So we define here the time step as 0.01 milliseconds. Then we define the total time, the duration of the simulation. In this case, the simulation would run for 100 milliseconds. Then we have the time vector, which creates an array of time points from 0 to 100 milliseconds in increments of 0.01 milliseconds. So we have 10,000 points in total, and it represents the simulation over time. This is the code. Then let's go to the third part, which is the external current injection. We define an array which stores the external current applied to the neuron at each time step. Initially, it is set to 0 at all time points. This means no current is applied. Between time steps 500 and 600, which corresponds to 5 to 6 milliseconds, a current of 10 microamperes is injected into the neuron. Imagine that as a brief pulse of external current. It will trigger a temporary change in the membrane potential. Let's go now to the initial conditions. We have an array V which stores the membrane potential at each time point. Initially, the membrane potential is set to V rest equals minus 65 millivolts at all time points. This means the neuron starts at its resting potential. It will change based on the injected and leak currents during the simulation. Now let's go to the simulation loop. This is the core of our simulation. To solve the equation, we use the Euler's method, which is used to numerically solve the differential equation governing the membrane potential. The equation we are solving are the membrane capacitance, the derivative of the voltage of the membrane potential to time, equals to the external current minus the leak current. Using the Euler's method, the membrane potential at time t is updated as follows. We have Vt equals V at the previous case plus the derivative, the time step, divided by the current multiplied by the external current minus the leak current. The leak current is the one that pulls the membrane potential towards the leak reversal potential. The membrane potential is updated by applying the current balance equation at each step, and it is influenced by both the external current and the leak current. And you can see here that the leak current is defined by the multiplication of GL with the voltage difference. Finally, we have the visualization, which generates the plot showing the membrane potential over time. The time points in milliseconds are plotted on the x-axis, and the corresponding membrane potentials are plotted in the y-axis. So we have a steady membrane potential at minus 65. Then we have a rise before stimulation. Then we have a rise in membrane potential during the current injection, and then we have the return to the resting potential. We are going to model it and fine-tune the parameters in order to see how each parameter affects the output of the simulation. We have now a simple model which shows how neurons respond to external current and how they return to equilibrium due to the leak channels. Thank you very much and see you in the classroom. Bye.